I'd like to introduce today's speaker, who's is Vadim Warshawsky. And Vadim started out his life in Russia and did biochemistry at Moscow State University, where he stayed for a couple of years as a junior scientist. And then Vadim came to the United States and came to the University of Wisconsin, and that's where our paths started to cross each other time and time again. Although when he was in Wisconsin, I didn't know him personally. I only knew him as the Russian family that was staying in the house of friends of mine while they were on sabbatical. So that's how we start off our relationship. So Vadim did some really fabulous work in the lab of Derek Bounds while he was at Wisconsin. And then from there, he went to Mass Eye and Ear as a faculty member, and rose through the ranks to obviously professor. And then he got recruited away from Harvard and to the beautiful campus of Duke University, where he is currently the senior scientific director in the Department of Ophthalmology. Vadim is very well known for his work on phototransduction processes, and in fact is one of the pioneers and has really defined all of the phototransduction steps in the photoreceptors themselves. And so part of that work he'll tell us about today. He and I then crossed paths at study section, where I was really impressed with how smart he is. And then most recently, his work has stemmed out into bipolar cells, where my lab is also doing a lot of work. And we had the opportunity to collaborate, and we just published a paper that came out last month in Neuron, so we're all very excited about that. So Vadim has also been awarded a lot of wonderful recognitions for his work. Jules and Doris Stein Research to Prevent Blindness Professorship, Senior Investigator for RPV, Alcon Award, and he is now the Helena Rubinstein Endowed Professor at Duke University. So today, Vadim is going to talk to us about timing is everything, regulation of G protein signaling in photoreceptors. Please welcome Vadim.
Thank you, Maureen. That was a lovely, embarrassing introduction.
Well, I was going to put up the cartoon, but I didn't do that.
All right. What I decided to do today is to tell you a story which started very, very long ago, and just to show you how an interesting question can develop, evolve, appear to be solved, appear to be unsolved again over a course of over 20 years. So it will be a little bit historical, but I'll show some unpublished data too. It will be a little bit of everything. And it's about timing of the photoresponse. Let me start by showing a couple of introductory slides. This is from Derek Bounds' slides collection, and I wish I knew who actually obtained this beautiful image. It's a frog retina. It's a scanning electron micrograph of a frog retina, and you can appreciate this amazing large cells, which are rods, and you can also see a couple cones. And although we have approximately the same ratio of rods and cones in our retinas, our photoreceptors are a little bit more comparable in size and in shape. Next slide shows what happens inside one of these cells when it's hit by light. So for the purpose of my talk today, it's just good enough to know that a photoreceptor has two parts. One part called the outer segment, which is a ciliary structure, and which is basically a light-sensitive antenna containing very large number of rhodopsin molecules, very tightly packed. So the light coming through this structure is actually never hit this way. It's hit from the bottom like this in real life. It could be very efficiently caught here. And there is the rest of the cell, ending with synaptic terminal, which talks to that very bipolar cells, which Maureen talked me into studying. I guess that was a study section, maybe. I don't know when it happened. That was a destiny. Rhodopsin is a G-protein coupled receptor, which is talking to a G-protein, to a trimeric G-protein called transducin. As all trimeric G-proteins, transducin has 3 subunits, alpha, beta, and gamma. And activation of transducin by rhodopsin consists of this exchange of GDP molecule for a GTP molecule on the alpha subunit of transducin. When that happens, transducin trimer falls apart into alpha subunit and to beta gamma complex. And in this particular system, and I will show you an example that could be different, but in this particular system, alpha subunit is an active entity which interacts with the downstream effector, phosphodiesterase of cyclic GMP. So this is the enzyme which hydrolyzes cyclic GMP when complexed with transducin. And what you can see in this cartoon, that this interaction occurs through this interface, the so-called gamma subunit of phosphodiesterase, which in inactive enzyme acts as a little molecular cork covering the active sites on the alpha and beta subunit of this enzyme. And then when transducin binds to phosphodiesterase, it binds directly to gamma. It moves it away physically from the active site, uncorks this enzyme, and this enzyme now hydrolyzes cyclic GMP. Now, all these events lead to the propagation of the photoresponse, such as this one recorded by Marie Burns from a mouse rod. And you can see that the photoresponse is kind of relatively rapid. It goes on rapidly, it goes off rapidly. So what is the virtue of having a G-protein mediating between a receptor, between the light-sensing molecule, and between the effector, which actually changes the concentration of cyclic GMP, causing this response? Now, this response is caused by cyclic GMP because this structure has ion channels which are directly gated by cyclic GMP. When cyclic GMP is hydrolyzed, channels are closed, and then what you see is a transient hyperpolarization of the cell caused by the closure of these channels. So back to G proteins. Why it's good to have G proteins here? There are 2 reasons. One reason is that during a course of a photoresponse like this, rhodopsin doesn't activate just one transducin. It activates somewhere between 15 and 25. And the signal becomes amplified by 15 to 25-fold. And that is very important to distinguish this signal from the electrical noise in this cell. And we won't talk anymore about that property of G proteins, but we'll concentrate for the rest on another property of this G protein to serve as a molecular switch. G protein would remain active here and in any other pathway as far as GTP remains bound to the alpha subunit. When GDP is hydrolyzed like this, then every activated component is returning into an active state. Trimer closes up. If there's no rhodopsin anymore being activated, then photoresponse recovers. And this general scheme explains how the photoresponse originates and how it is turned off. So about 20-something years ago, when I was a postdoc, I was really bothered by this question. So whenever I was able to purify transducin, put it in a test tube, and measure GDPase activity of this enzyme, the rate of hydrolysis was more like 1 turnover per minute or something like that, rather than occurring on this time scale where you can appreciate by about 0.2 seconds, this response is almost completely gone. So there was some problem, some quantitative problem, which bothered me. And we started designing experiments looking whether there is something in this cell which can regulate the rate at which transducin can hydrolyze GTP. And the first very productive experiment in this direction is right here. So what we did was essentially very simple. We purified photoreceptor membranes in such a way that they retained transducin, but they were deprived of its effector phosphodiesterase, and we measured GTPase activity, and it was fairly slow. And then we added this effector back, phosphodiesterase, which we purified and separated. And then GTPase activity became very fast. And then we took a recombinant gamma subunit, this little quark, this little interface between the two, edited back, and GTPase activity became very fast once again. And of course, we were extremely happy at that moment because, for first, it seems like we answered the question. We understood that this interaction of G protein with its effector regulates its GTPase activity, and the puzzle is solved. We're also very happy at the moment because it was the first case ever to show that the rate at which G proteins inactivate could be regulated by any other molecule whatsoever. And perhaps we were even more happy about that. The problems started coming up very soon because some people were able and some others were not able to reproduce our data. And when we investigated what is going on, we also figured out that we cannot always reproduce this effect. And in particular, if instead of taking photoreceptor membranes, we would take purified rhodopsin, put it into membrane vesicles, we'll take purified transducin, and we'll add purified phosphodiesterase, well, nothing particularly interesting will happen with the rate of GTPase activity we measured. And we realized that this membrane had something else which cooperated with this gamma subunit in regulation of transducin GTPase. 6 years have passed. We were close to purifying that thing biochemically, but never did. And it turned out that there's a big class of molecules, which wasn't known at the time of this experiment, called regulators of G protein signaling proteins, or RGS proteins. And all of them interact with various alpha subunits of G proteins, serving as a GTPase activating entities in a variety of pathways. In fact, I don't think there is any cell of our body which does not have one or another G protein. It certainly wouldn't be transducin anywhere away from our eyes, but there will be 25 cousins of transducins, and there will be one of or more than RGS proteins acting there, negotiating at what time these G proteins should be inactivated. In this particular cell, which we're studying photoreceptors, it turned out that this role of a GTPase activating protein belongs to the 9th member of RGS family, RGS9, which is existing as a constitutive dimer with a beta subunit, which has a name of G beta 5. This was shown by Ted Wenzel, that this number 9 member of the family works in this particular pathway. My laboratory identified G beta 5 as a member of this complex. So the story became a bit more complicated. We still know that this gamma subunit is important, but now we have this 2 subunit protein in addition to that, and we need to explain who is doing what. Because remember, there was no much acceleration without gamma as well, when RGS was present without gamma. So let me start with the gamma. And in principle, you can envision 2 different scenarios. One is that binding of transducin to gamma changes the affinity for this complex. Right? So it's kind of an affinity enhancer in this system. Another option is that RGS9 is just a bad RGS protein, and it needs some pieces from gamma to make up for some deficiency in its own structure. Right? You can distinguish these options in experiments, of which I will show you the most simple one. In this particular case, we have a recombinant RGS9 G beta 5 complex attached to agarose beads, and we're adding to this complex alpha subunit alone or alpha subunit in a complex with this gamma protein. And you can appreciate that the retention of this alpha subunit on the beads is significantly better than the retention of free alpha subunit. Right? Kinetic experiments show that what you observe here is about 25-fold difference in the affinity between this complex and this molecule. Right, so let's try to make some sense out of it before we move on with discussing other parts of this complex. So I'm showing this slide. Let's just think what transducin does and how it works. So here is the alpha subunit of transducin freshly activated by rhodopsin. Now, it needs to fulfill two tasks which are not necessarily well compatible. Task number one is to go very rapidly, find phosphodiesterase, activate it, and not lose any signal en route. Task number two, our vision is rapid. The whole complex has to be inactivated very rapidly as well through interaction with an RGS protein. Now, if you think about it, making RGS binding to this complex contingent on G protein forming this complex with effector and activating the effector first brings some very nice order into the whole process, right? You activate your target first, you inactivate second, right? So nothing is left to chance, no information is missed in a route. Makes sense, and in fact, we felt that it makes so much sense that it would be interesting to see if some other signaling pathways may have proteins like PDE gamma or maybe protein domains like PDE gamma that might fulfill a similar function. And that brought us to consider a different splice isoform of RGS9. So RGS9 is expressed both in the retina and in striatum, and it comes in two flavors. Number one, of course, as always, is from vision. This is the hour guy. And number two is expressed in striatum. It's involved in the regulation of motor behavior, in some regulation of some pharmacological issues. For instance, mice lacking RGS9 are cocaine addicts. Apparently, people are not, although I think there are only 5 known. Maybe not enough statistics, or maybe not enough disclosure. And what we found particularly amazing about this protein, kind of strangely overlooked for a while, is that this difference here in the C-terminus of this striatal isoform looks awfully like PDE gamma. It has a very similar cationic-anionic transition at the boundary of one exon, and most importantly, somewhere here at the C-terminus, there is identical sequence of about 6 amino acids, and you say, you know, coincidences are happen. But an interesting thing about this particular sequence is that if you take PDE gamma and you mutate this particular tryptophan to alanine, this single amino acid substitution is depriving PDE gamma from the ability to cooperate with RGS9 interacting with transducin. We published a paper about that with Stephen Sung and others. So we were very excited about what we've seen, and we conducted experiments similar to those which I'll show. So once again, I wouldn't torture you with any enzyme kinetics, and instead we'll show one more pull-down experiment. So what you have here, you have each splice isoform with G-beta 5, they're all dimers, attached to agarose beads. And now to these beads, we are adding a G protein called GO. Now remember that transducin is specific for the photoreceptors. The striatum does not have transducin expressed, but it has G protein called GO, which is a very close relative of transducin. And in fact, even rhodopsin can activate GO as well as it can activate transducin. So we're using GO this time. And once again, just like in the case of transducin, PDE gamma is enhancing the affinity of GO to RGS9. 1, right? Same protein we were discussing before. Now, this interesting splice isoform in striatum, which has something which looks like PDE gamma built into its own structure, right? Now the affinity for GO without PDE gamma is about as good as in this case with PDE gamma. You can add PDE gamma, not much is happening in this case. If you add very much, it will start competing. But Yes.
Clarify something for me, because you initially started out by saying that that you started down this pathway because it took 0.2 seconds to generate the photoreceptor signal, and when you observed chemically, it was obviously much longer. How is this explaining the time issue? Is it just because the number of molecules that are being produced that is allowing?
No, it is the rate at which the molecule is inactivated becomes very rapid through this interaction.
And it's then becoming activated again?
If you keep light on, yes, but if you just have a single flash of light, rhodopsin inactivates on a time which we now believe is about 50 milliseconds. So during that time course, you activate about 20 of these G proteins, and then the rate at which your photoresponse will go off will be primarily determined how long they will leave with GTP. So you let them leave, and I will show some data maybe in a few minutes how it actually looks in electrophysiological recordings. But if you leave with GTP long, the response will stay long. And if you are adding this fellow, it just cuts off very rapidly.
Is adding PDA gamma to RGDS92 mutated version, that proportion, is it actually
Right. In this example we're here now, we are just having a PDA gamma built into the structure of this fellow. And let me try to speculate why it would be good in striatum. So what happens with many G proteins is that the signal then conveyed to downstream targets is not conveyed by alpha subunit, but is conveyed through beta gamma subunits. In fact, perhaps this is the most original evolutionary prototypic way in which G proteins are functioning. A receptor, and in this case it may be mu-opioid, it may be D2 dopamine perhaps, would activate this protein GO, and in this particular example, the result of this activation will be beta gamma releasing from alpha, interacting with the downstream effector, for example, this potassium channel, just for the sake of an example. What happens within alpha subunit in this pathway, it wouldn't activate anything downstream, but instead it will serve as a timer in this system. The entire function of alpha would be to time the duration of this event by interacting with an RGS protein, and the rate of this interaction will determine how long this channel will remain open, right? So in this case, when beta gamma subunits are playing the role of activating effector, it seems to be rational, at least, I mean, to expect that the high affinity element, this PDA gamma-like domain, would be positioned right on the RGS. So this interaction could be efficient, rapid, and the channel would not stay active too long, right? So that's kind of a lesson which we learned from this system, starting with vision and going further to the brain. Let's go back to why this protein is so large, why there are two subunits, if there is any functional role of this protein to be a dimer. The crystal structure is solved by John Sondek at UNC, and it looks like this, but I feel that this would be probably better, a little bit friendlier image to discuss. And what you see here is that RGS9 polypeptide chain is pretty long. Only about a quarter of it's taken by this RGS domain, which immediately interacts with G protein and tells it to hydrolyze GTP. Then you have this block of molecules, this domain called G protein gamma subunit-like domain, serving to attach G beta 5. Right? This is the second subunit. And this subunit is serving as a divider of a sorts between this catalytic part of RGS and the secondary part of RGS, and we'll discuss what that part does in a little bit. But there is a big divider here, and all the function, the ability to tell G protein when to inactivate, is confined to this orange domain. Right? For those of you who are familiar with the G protein structures, I can stress that this whole module looks awfully much as a G beta gamma subunit of G proteins, although in this case it's just a part of the RGS structure. So the next slide illustrates our experimental strategy. And what we decided to do is just to imagine that this whole molecule is a kind of a Tinker toy box. We have these little elements, and now we can build the different molecules as recombinant proteins in which we combine this catalytic domain of RGS9 with other little parts. And I will skip many experiments and will show you perhaps the most interesting one. And again, the same idea of pull-down assay. So you assess the affinities between the molecules you are studying back to transducin, right? And if you have this domain alone, it does have some affinity for transducin. But this affinity really is not regulated much by PDE gamma. You can't really appreciate much of a difference here. Right? So we have relatively high affinity, but we don't have any recognition selectivity for this entity. If we add this module of G beta 5 subunit with this gamma-like domain, this is just one module. You can't add them separately. They don't exist without one another. What you would see a big reduction in the affinity of this construct to G protein. And once again, you wouldn't see much of a difference with and without PDE gamma. But then when you restore the protein to its entirety, and it turns out that every single other little piece matters upon this restoration, you will keep affinity low for the wrong target, for the free alpha subunit, and you will enhance affinity very much for the right target, for transducin bound to PDE gamma. So let me repeat that in a little bit different didactic way. So this is a little movie, you will see, little cartoon, in which I depicted a relative affinity between proteins by relative distance. And what this first frame is illustrating is that when you just have this domain alone, that is the domain which tells transducin to hydrolyze GDP, then in fact there is not much difference in the affinity for the right target, this one, effector complex, and the wrong target, free activated transducin, right? When we add this one, then ouch will happen. And this particular construct doesn't really like anyone, right or wrong. And then we'll start adding little other pieces, and every piece will matter here, every missing part. And then this addition will restore the affinity for the right target, keeping the affinity for the wrong target pretty much intact, low. Very strange mechanism. Why couldn't you just design this little orange piece so it works the way? I have no idea, but that's how it works. I guess to keep us scientists busy. To complicate the story even further, so all the catalysis is located here, but the N-terminus, this other part of RGS molecule, is very important for its membrane attachment. And the membrane attachment occurs through the anchor protein called R9AP, RGS9 anchor protein. The protein was first identified by Ted Wenzel, although there's another very interesting story I will tell you a few minutes later. And it turned out, what we found, that this binding has very significant consequences for the ability of this molecule to activate transducin GDPase. So you can take this molecule alone, and it can recognize right and wrong, but the absolute rate at which GDP would be hydrolyzed in transducin would be still somehow low, just a bit slower than kind of once per second. That's not bad at all. But it's not exactly as fast as that photoresponse was inactivated. When you anchor this complex like this, then the activity goes up very significantly. You can see this time about like 7 times per second. That's enough. That's good enough to inactivate photoresponse, and it's real time scale. And we think we understand how it works. And understanding the structure of this complex really shows that because catalysis and anchorage are so much separated from one another, I think the whole idea of this acceleration consists of this protein being on a membrane surface, meeting transducin on the membrane surface, and just providing good thermodynamic environments so they meet each other with the right kind of a sites, having not too much freedoms of rotations when they interact. I think that's how we believe this mechanism works. Well, it turned out that R9EP has a couple of other very important functions, and one of which was revealed when we expressed in the mice, instead of their natural full-length RGS9 lacking this domain serving for R9EP anchorage, right? So you have the molecule which is catalytically active but lacks this piece. What happens? Well, nothing much happens with the structure of this retina. This is not one of mutations causing degeneration. But some interesting things happened regarding this protein's localization. So that's just one more control showing that this mutant construct is expressed at about the same amount as a wild-type protein, and that the R9EP in both situations, the anchor protein is pretty much normal. But when we conducted immunohistochemistry on the retinas from these mice, we found that despite normal localization of the anchor, the localization of RGS9 mutant is very wrong. We don't see much in the outer segment. All immunostaining is mislocalized to the other parts of the cell. So it seems like the interaction with this anchor is very critical to bringing this protein to the outer segment or just for keeping it in the outer segment. And let's see now what happens if you knock out R9EP instead, right? Would you see exactly the same result? This is probably a good time to tell you a little bit of a story. So, well, it turned out that people who really discovered R9EP was Jim Hudspeth. When they were looking for markers, unique protein markers in hair cells in the ear. And Jim had a postdoc by the name of Stefan Heller who worked at the Mass Eye and Ear in the ear department. I think he's in Stanford now. And they decided to make a knockout, R9EP knockout in the mouse. By the time they made a knockout, it turned out that this is a chicken story about R9EP being present in the hair cell. Mice don't have R9EP expressed in the hair cells. So they were kind of trying to figure out what to do with this mouse. They stained every potential tissue they could, and then came to my office. His student came to my office, said, well, look, I mean, we have this strange protein. It's kind of a very sad story. We made a knockout. We don't know what to do with that. But it stains outer segments of photoreceptors among all tissues we could ever find. And R9EP was not published at the time by Ted, but we knew the sequence already. It was like, weeks before the publication. He showed it already at a meeting, so we knew what it is. I don't know what happened to me, and I just kind of, opened that sequence and asked this, and then I've seen the most amazing scene, a petrified postdoc. So anyway, that was kind of nice. We had a knockout even before publication, and knockout actually yielded a very interesting result. So let's knock it out. Morphology was very nice once again, which is always very rewarding when you're studying functions of the cells that like to degenerate for many reasons. And then you can also see that knockout was real. There was no any R9EP expressed in these retinas. But then when we looked at RGS9, we figured out that it's completely knocked out as well. And it's knocked out despite the fact that mRNA levels for RGS9 were perfectly normal in the knockout. The whole regulation occurred on post-translational level. Well, it turned out that R9EP plays a very important role in protecting RGS9 from intracellular proteolysis, and ultimately by playing this role, it is really the protein which sends the temporal resolution of a photoreceptor cell. This is really the protein which expression decides what would be the time constant of a given receptor. And indeed, we know that rods are a bit slower than cones. Cones work a little bit faster. And there's some very nice evidence that cones contain a few times more R9EP than rods. And clearly, this is one of the important contributing factors to that difference. So to summarize, we have a very strange complex with a lot of parts, and basically neither one of them is disposable. Some of them are responsible for catalysis, some of them are modulate catalysis further, some of them are responsible for allowing this molecule to distinguish between right and wrong substrate. And R9EP in particular is a very interesting beast because not only it serves as a membrane anchor, you know, big deal, you could have lipidated this molecule, would be the same result. Also, it's a very savvy protein, changing the catalytic activity of this complex, targeting it to the right place, determining its intracellular level. And for the second time, we became very curious, what if there's something like R9EP? Like you already know, R9EP is not expressed in a hair cell. Now, turn out it's not expressed pretty much anywhere but in photoreceptors and bipolar cells. But about 11 months of conducting immunoprecipitations from mushed brains with antibodies recognizing RGS9's striatal homologue, RGS92, yielded this gel. And with this is something which I find totally amazing about mass spectrometry. You know, 11 months of monkeying with this gel and about 2 hours of mass spectrometry lead you to discovery of completely new protein, which we called R7BP. The name is kind of bad, I know, but I mean, we kind of thought about, didn't come with anything else. So RGS9 belongs to subfamily, which is called R7 RGS proteins, and R7BP stands for RGS type 7 binding protein, right? And this fellow turned out to look extraordinarily similar to R9EP by its structure. It doesn't have a transmembrane domain. It has multiple palmethylations. It's also membrane bound. And also both of them look somehow like syntaxins. So for those of you who are familiar with synaptic transmission and vesicular fusion, you know, syntaxins are protein, very important for vesicular fusion. And we have no idea why these proteins look like syntaxins because we really can't assign any function which would be somehow related to membrane fusion, but they look like it. Now, my postdoc, Kirill Martymyanov, who is now at Scripps, took that part of the project with him, and he and some other people showed that indeed in the brain and in bipolar cells of the retina, this protein plays very similar role to the role of R9EP in photoreceptors. It does anchor the members of RGS family are dendrites of multiple neurons. It does regulate their expression levels. It's actually a very important protein, overall speaking, perhaps more ubiquitous protein than R9EP. But this lead through the function of R9EP was very critical for R7BP discovery. Let's get back to vision. And let us talk about something different. Let's talk about what actually happens with vision if you don't have a rapid inactivation of G protein by this amazing complex, right? So you can do this experiment. This is from a classical work between Jason Chen and Marie Burns, and they both were postdocs with Mel Simon and Dennis Baylor at the time, kind of one of the most cherished papers in the field. This is a phenotype of RGS9 knockout, right? You knock out RGS9, and the photoreceptor, instead of being fast, turns into a slow one. You can appreciate that the rising phase is not affected very much. It's decay phase because the activation of G protein is normal. It's an activation phase, which is now slower. Now, going back to your question, I actually still don't understand why it's 2.5 seconds instead of like closer to a minute. We suspect that maybe there is something else, some hidden RGS protein, which is not very prominent in the wild type, and that's why it's a bit faster than you would expect. But that's kind of one of the, you know, little remaining mysteries in the field. And this is, of course, very nice and very predictable, but one problem with mice is that they kind of have a very hard time telling us what exactly is happening with them when they switch from responses like this to responses like that. And it's kind of interesting to appreciate what exactly would happen with our vision when the responses go on normally but are slow. And that brings us to Ted Dreischer, who was a colleague at the Mass Pioneer at the time, got extremely interested in this question and spent quite a few years trying to find people who have impaired function of RGS-9. And he and his colleague in the Netherlands, by the name Art Koopman, found 5 people, apparently unrelated, but obviously they're related somewhere in the past, which all have this mutation in RGS-9 protein right here. Right, so what you see here is a crystal structure of the catalytic domain, the orange fellow on my cartoons. And you can appreciate that the whole domain is just a bunch of alpha helices, like, you know, like a bowl of spaghetti. But here, at the entry point and exit points, there is this collar of hydrophobic residues, 5 of them, which are keeping the whole structure together. And the most hydrophobic of all hydrophobics here is this tryptophan at position 299. And in these people, it's substituted for arginine, which is not hydrophobic. It's very hydrophilic. And indeed, when we expressed, that was kind of our contribution to that study, this protein as a recombinant protein with this mutation, we pretty much literally could see the protein falling out of solution in front of our eyes. And what I've stated in the solution was practically inactive in its ability to help transduce into hydrolyzed GTP, right? So for any practical reason, this is almost the same as RGS-9 knockout. What do these people complain? First of all, photophobia, because their responses to light are staggering on the top of one another, right? So they really can be blinded by bright light much easier than people who recover their responses very rapidly. But perhaps the most amazing phenotype came kind of in the 11th hour of their investigation when they were subjected to dynamic acuity tests, right? What is dynamic acuity test? It's like acuity test, like, you know, I'm testing you now to see the letter E on this screen. It will be important that the letter would be of a low contrast to reveal their phenotype. And now this is a dynamic acuity test. Now you're asked to tell what letter is moving through the screen. Well, these people, apart from not liking bright screens, these people had no impairment in seeing a stationary letter, no matter what contrast this letter was. But when you moved letter like that through the screen, it was very interesting. They were presented this low contrast letter, and they're very polite people. They will look at the screen, they will sit there for a while, and they say, "Doctor, you forgot about me." I find it fascinating because, you know, before I knew that, I would have asked a question in the classroom and I will say, you know, so what do you think? What will happen if photo responses are not inactivated? And a smart student will always raise a hand and say, oh, we will have kind of a stroboscopic kind of overlaying of images like these multiple photographs on an athlete in a job. Kind of, you know, there will be smudges, you know, we'll have multiple images of same people as we move the head. Well, that's kind of what physiology suggests, but it looks like, you know, our visual processing is so savvy that this is probably not an optimal situation. Like, you know, I kind of, you know, I look at Marie and then I look at Henry and I kind of don't know who I'm talking to. I could put myself in trouble, right? So the safe bet well, the safe bet would be not to see any one of you before I'm stationary enough and I know exactly who is in front of me. At least that's how our visual system works. A pretty fascinating story.
Is that dependent upon the level of visual acuity? In other words, with small letters?
I wish, you know, I wish we tried to follow up. You know, Ted is geneticist. And what we really needed is a really good first-class psychophysicist to bring these people back, stop torturing them for the ARGs. Because actually, it's very hard to get them, because what happens is they go, they're tortured with the ARGs for a whole day, they hate it, and then they're told, thank you very much, we can't actually help your condition. You won't be able to play ball game no matter what I tell you. So they actually don't like to be studied, but I guess it's much easier to team up. And we tried to do it with Bob Barlow. Bob passed away a couple years ago, and I actually didn't regain my efforts. I'm not a psychophysicist. It's a little bit far. You know, I can only inspire people, but I can't really analyze these patients. But I think I think these patients are a gold mine, frankly. But that's pretty much, as today, that's pretty much all we need. There's some more of them identified in the world, one in Singapore, but again, everything you see are ERGs. Very characteristic.
Can they drive?
Yes. Believe it or not, some of them are driving. They can't play kind of fast ball games when the environment is of high contrast. So I suspect that they probably would avoid driving at night. And neither one of them, as far as we know, is a cocaine addict, which is kind of interesting because the mice are. Particularly being in the Netherlands, that's kind of,